Welcome to Vaisumit. Today we'll learn about chromatography. So here you can see chroma in Greek means color and graphene means writing. Actually Russian botanist Soet, who is also called the father of chromatography, he used some techniques to separate some plant pigments like chlorophylls, xanthophylls, etc. And he did some color bands. That's why it is called as chromatography. Now why we will do it? For the physical method for separation of compounds. It is physical method for separation of compounds. Like for protein and peptide purification, so we actually employ it for peptide purification or any other purpose to separate the compounds. Now two main factors of chromatography is one mobile phase and one stationary phase. The mobile phase is what? Mixture of substances to be fractionally dissolved in a liquid or gas fluid. And stationary phase is resultant solution is percolated through a column consisting of a porous solid matrix. Now the main idea is there will be different degree of migration of the stress of the mobile phase along the column or any plane. So on the basis of that migration, one can migrate it through the column faster and then the other will be later. So we can separate those compound with some time or maybe some size or maybe some affinity. So we'll look after this with forward with this. Now on the basis of mobile phase that will be migrated through the column or any plane, it can be divided as liquid chromatography or gas chromatography. So in liquid chromatography, the mobile phase will be liquid and here the mobile phase will be gas. Now also it can be divided into column and planar chromatography. So in this video we will learn just the basics of the chromatography. Now in planar chromatography what happens? The stationary phase will be present in a plane. Okay. And in column chromatography the stationary bed is within a tube. So we will look with a diagram. Here in this diagram you can see an example of column chromatography. So what is happening here? Here there is solvent, that is mobile phase. Okay. Now in this solvent there will be stationary phase that will be suspended in the solvent. Now here the fresh solvent is getting introduced here, and we have to separate two different things, that is A and B. Let's say I have denoted A as in blue and B in red. So here you can see initially you can have some very little difference between them, the very little difference in band between them. With the time you can see B is going faster, right? And here the solvent will be flowing out, right? Now with the time here you can see the B is emerging, right? B here is emerging. So after a few time B will be gone from this column. Now there will be only A and maybe then A will be emerging out, right? So this is the basis of column chromatography. So chromatography can be divided in many groups, like in the gas chromatography. Now if you are taking in consideration the stationary phase, then it can be gas chromatography can be divided into two, that is gas solid, and another is gas liquid. Similarly the liquid is also can be divided into two subgroups, like liquid solid and liquid chromatography. Now the chromatographic techniques use several types of interaction to separate solids. It's not only the size or something. There will be many things like size, or it can be affinity, or it can be affinity for some ions. Okay. Now based on these types of interaction, there can be so many chromatographic modes. Okay, like adsorption, size exclusion, ion exchange. In our next video we will learn about the details of this size exclusion or ion exchange or affinity chromatography, these things. So here in this video we can just uh, know the basics of the chromatography, why it is used and why and how it is used in a simple way. Remember one thing, chromatography actually represents a technique. Okay, now what is chromatograph? Now chromatography is the system for performing chromatography. Now another similar term comes in this topic, that is chromatogram. This is the record or the result which will be detected by the detector, right? So chromatography, this is the technique. Chromatograph, this is the system, and chromatogram, that is recorded. Okay, so for peptide purification or any protein purification, like the same thing actually. So we will do the chromatography due to some based on many things. It can be divided into liquid chromatography or gas chromatography. It is based on the mobile phase. Now if I am taking about the stationary phase into consideration, then liquid phase can be liquid solid, liquid liquid, gas can be gas solid, gas liquid. Based on some other things, it can be column or planar, that is the stationary phases position. Stationary phase can be in the plane or can be in a bed like this. It can be separated like this, and there are many more like adsorption, size exclusion, or exchange. So we'll talk in details in our next video. So I hope you understand the basics of the chromatography, but don't worry, we will look in details with upcoming videos that will be very helpful. Thank you.